when I started. Okay, so welcome everyone to Super Steam Saturday on a very cold, chilly January Saturday. And it is my great honor and pleasure to introduce Dr. Emily Thompson. And I'm going to let her take over from here. And please enjoy.
Thanks. So hello, everyone. You can call me Emily or Dr. T. And today we're going to talk a little bit about dogs. And then, well, I like stuffed animals. So I got that from my kids' room. And then a little bit about wolves and how we got from dogs to wolves. So I'm going to share my screen. And while I'm sharing my screen, I'd love to see in the chat maybe what grade you're in, and if you have a dog. Maybe that's what your interest is in. And Susan, do you think you could read those to me as I'm pulling up the slides? Oh, Susan, I don't think I can hear you.
Oh, I'm sorry. Okay, so Max, we have Max Lawson with us today.
Yeah, it's actually Lawson.
Your brother will be joining.
Max will be joining soon.
Oh, okay. Okay, so we're gonna have two brothers. So Max and Lawson, right?
Yep, that's right.
Okay, so Max and Lawson are with us. And Max, how old is Max Lawson?
He is 12.
Okay,
I'm 11.
Max.
We're
Okay.
11 months apart.
Okay, 12. And then Lawson, I forget your age.
Oh, 11.
Okay, is 11. Okay.
Wonderful.
And Vicky is with us from MSU. And
welcome.
she is pretty much joining most of our Super Steam Saturdays, I believe the virtual ones, right, Vicky?
Yes,
Yes.
maybe some in person
Yep.
if my schedule allows. Yeah.
Wonderful. Well, welcome everyone. Today we're going to look at Fido and the Gray Wolf, and we're going to look at a few topics here, which are on the next slide. So the three things we're going to go over are a little bit on the genetics of dog coat color. Second topic is going to be saving the wolves of Isle Royal, which is one of our national parks in the state of Michigan. And then I have recorded a demonstration of how to extract DNA, which would be something you could do at home. And we'll be dropping the instructions in the chat when we get to that section. So one question is, what's America's favorite dog? And I just got a chat about, oh, you have a dog, an Irish cream lab. Oh, fabulous. We will be talking about labs. So what's America's favorite dog? And you could write that in the chat. What do you think it is? What are among the favorite dogs? All right, Golden Retriever is one of the guesses, and that's going to be correct. What else have we got? A Black Lab. And I think we've got another guess coming. People usually guess their own dogs. All right. A Pit Bull or Lab Retriever. Okay. And so this is what the American Kennel Club says are the most popular dogs: a Lab, a Yorkshire Terrier, German Shepherd, Golden Retriever, Beagle, Dachshund, Boxer, Poodle, Shih Tzu, and Miniature Schnauzer. So that's what they say. And they forgot mutts. Mutts are popular. That's what we had. All right, so I want to tell you a little story. And you might have heard this story because there's a very famous early reader. You might have read it in first or second grade. And this is the story of Balto, but also the dog Togo. And these are sled dog heroes from 1925. And shown here, specifically right here, let me get my laser pointer. Right here on the left is the hero dog Togo. So our story is that in 1925 in Nome, Alaska, 2 children got diphtheria, which is a deadly disease. The treatment is an antitoxin serum, and it wasn't widely available. So they didn't have any in Nome. And they knew that the diphtheria was going to spread throughout the children in town because it's so communicable. So here is Nome, Alaska, and the place where the antitoxin serum was Anchorage. So they arranged with people in Anchorage to put a lot of doses onto a train because there was a blizzard going on and they couldn't fly airplanes in 1925 in a blizzard. And the train started off towards Nome, Alaska, but it got stuck in Nenana and because the blizzard snowed it in. And because of the blizzard, the only way to get it from Nenana to Nome was dog sled. The Nome officials organized, I think it was 20 sled dogs to be at way stations to pick up the antitoxin and deliver it one to the other. And so one of the first legs involved a musher or sled driver by the name of Leonard Cepolo. And his dog, Togo, was one of the heroes. So here's Sepola and the dog Togo. They managed to do 264 miles. So they had to, overall, there was 700 miles that had to be traveled. And they did almost a third when most teams were only able to drive 31 miles. So in other words, this dog was fleet of foot and knew where he was going, no lollygagging around. So that was one hero dog. And then the dog that was supposed to do the next to last part of the relay was Balto, and his driver was Kaasen, shown here. These dogs actually were from the same kennel, but they had different mushers driving them. Balto was supposed to drive the next to last session. And so the blizzard was howling and they had 30-mile-an-hour winds, and it was below minus 35 below. So Kaasen drove the dogs on his leg of the journey, and there was no one to meet him. So they just continued. And so he drove 20 hours straight, and they made it all the way to Nome. They were expecting the whole team to take 15 days to get from the place where the antitoxin was stuck on the train to Nome. It took them only 5 days because of the heroism of these two mushing teams. In honor of this miraculous delivery of the antitoxin, which did save the children of Nome, Alaska, the Iditarod is a race you might have heard of that goes actually from Anchorage to Nome. And there's a winner every year. So here's a sled dog team. This one is led by Susan Butcher, and she was a 4-time winner of the Iditarod. So that's pretty cool. And they've put up statues to both dogs. Balto has a statue in New York City because this run captured the attention of the whole United States. And so Balto's statue is in New York City in Central Park, and Togo's statue is in Seward Park in New York City. And there's a little bit of genetics here. Balto was not a pure Siberian Husky, and so he was not considered to be most people wouldn't have thought he would be a good sled dog, although he ended up being tremendous. So he was 39% Siberian Husky, 18% Greenland dog, 17% Chinese village dog, 6% Samoyed, and 4% Alaskan Malamute. That's pretty amazing. So I was trying to get you in the mood to study dog coat color here. And so dog coat color comes from 2 different pigments or colors made in the epidermis or skin of dogs. So one is a black pigment or color called eumelanin, and it affects coats and eyes and nose color. So you can see over here, the black puppy has a lot of eumelanin. There's also a red pigment, and that's pheomelanin, and that affects coat color only. So you can see over here, this golden lab has the pheomelanin influencing coat color, but then the black pigment, eumelanin, is giving the dog the black nose and the black eyes here. Most dogs have both pigments, except for white dogs. And so dog coat color is pretty complicated. There are all these genes involving coat color. So A, B, D, E, G, H, K, M, S, and T. I do not know all of these. I'm not an expert on these. I teach genetics, but I know a couple of these genes. And so I'm going to tell you about a couple of them. So these genes all together in the dog will lead to a lot of dog coat colors. This includes the apricot color. Does that really look apricot to you? I don't know. That's what the American Kennel Club calls apricot. Here's one called lilac. It's kind of purple. So that's pretty cool. Here's a silver one. That's pretty awesome. And then they have a number of different patterns of coats. Here's some that are called striped and brindle. And they have a dog even that's called a phantom. Here are spotted ones. So all those genes make all the colors and color patterns. So where do we start? The DNA is on chromosomes. So DNA is a molecule. It's on the chromosomes, and the chromosomes are in the nucleus in the cell. So here we're looking at a nucleus. The chromosomes are these X's, and it's in the cell. Dogs have DNA on chromosomes, and humans have DNA on chromosomes. Plants have DNA on chromosomes, and so on. So dogs actually have 78 chromosomes, and here's a chromosomal spread of dog, and the dog's Latin name is Canis familiaris. Humans have 46 chromosomes. Do any of you know our Latin name? Go ahead, Lawson.
Homo sapien.
There you go. All right. All right. So Canis familiaris means dog that we all know, and Homo sapiens means wise man in Latin. All right. When we're talking about genes and genetics, here are two chromosomes. In this case, they're not looking like X's. They just drew them as lines in this case. And there are different versions of genes. So here we're going to have two different versions. We're going to talk about the B locus. So we have capital B and lowercase b, and you might have studied these in school. You have each dog has a ma and a pa, and each of those parents gives one copy of gene B to them. So, and gene big B, little b controls whether the lab is black or the so-called chocolate. So if you have big B, then you're a black lab. If you're big B, little b, you're a black lab. If you're little b, big B, you're a black lab. And so basically we could say, as long as you have at least one big B, you're going to be a black lab when we look just at the B. Over here, we have a chocolate lab, and they have as the genes they got from ma and pa, little b and little b, and that makes them a chocolate lab. All right, so there's more. When we do, oh, sorry, there's a question. Go ahead.
Oh, I was just gonna say, isn't it the two smaller bees are recessive and it's that the large bee is dominant, and as long as there's a dominant, the recessive is not gonna show, right?
Correct, in the phenotype. Exactly. So you folks are ahead of the game here. So we're going to do a little Punnett square, and it sounds like you might have done this in school or at home or something. So the Punnett square allows us to look at the cross. We're going to look at three crosses, the first one, the second one, and the third one. So we've got pa on the left and ma on the right. You can tell I just copied the picture over. And below, we're looking at the chances for getting the puppies. And I'm going to grab myself a pen here. Let's see. All right. So pa has both genes are the same. Pa is big B, big B. he has half a chance of donating Big B, the first one, and he has half a chance of donating the second one when he's making puppies. And over here, the female is also Big B, and she has half a chance of donating the first Big B. She has half a chance of donating the second little B. And then we can, maybe I'm going to try typing because I got some crazy smearing here. Let's see. Okay, never mind. All right. So when we look at the genotypes, the genes that are donated to the puppies, all of these puppies have the same genes. They get a Big B from Pa and a Big B from Ma. So they all are Big B, and they're all are black labs as shown in the picture here. All right, so far so good. Next, we have Pa is a black lab and we have Ma is a chocolate lab. So when we do the Punnett square, Pa can donate either the first Big B or the second Big B, and Ma can donate the first little b or the second little b. And as Lawson said, if because Big B is dominant, if the puppies have one Big B, then they will be a black lab. And we see that when we have a cross with Big B by little b, all the puppies are still black labs. Okay, so the lowercase little b is recessive and it's hidden in the phenotype. All right, so next we have two black labs, let's say from the previous experiment here, and they are, they have the alleles Big B and little b each. So Pa has a half a chance of donating Big B, half a chance of donating little b. So does Ma. And when we write down the alleles that each dog gets, the first one, this means that there's a quarter chance that the animal is Big B, and looks like a black lab. Then there's a 2/4 chance that the animal is Big B, little b. They're still black labs, but they have different alleles. And then there's a 1/4 chance that the animal is little b. I think I'm going to write it down here. And as a chocolate lab. So these Punnett squares allow us to determine what the cross is going to be. So are there any questions or comments so far? We're going to add a gene. You folks are doing so well. All right, let me erase everything. Okay. All right, so our summary is that gene capital B gives the black color. It's dominant over little b. That is chocolate, and that gives the chocolate lab, and it's recessive. So we could say Big B, little b, or little b, Big B gives black. And when we write these two in a different order, it just means which parent is giving the little b. In the first case, Ma is giving the little b, and in the second case, Pa is giving the little b, because sometimes it matters. So we could also summarize this by saying, as long as you have at least one Big B dash gives the black color. Little b gives the chocolate color. And so you have to have both parents giving you little b to have the chocolate color. All right, so we summarized that before. And so let's investigate a little further. Big E, little e controls whether a lab has a yellow color or another coat color. So if you have little e, you are yellow. If you have at least one Big E, you are not yellow. So this animal is little e, and it can have any arrangement at B. Over here, both of these animals, to have either a black or a chocolate color, have to have at least one Big E. So this one is Big E, little e, and then it could be Big B, it could be Big B, little b, or little b, Big B. And this animal that is chocolate lab, let's see, trying to get my, laser pointer. Okay, this one that's chocolate lab has one big E, so it can be not yellow, and it has to be little b to be chocolate. So we've kind of got the idea for how you can get 3 different coat colors in labs. All right, so let's write that down in case it's easier to learn it by looking at it. The little e gives the yellow, big E dash gives not yellow, and that allows us to have a black lab or a chocolate lab. Okay, any questions so far? All right, you all are doing great with the dog's coat color here. All right, and so what about other coats? Like what about white coats? And in this case, other genes are involved. They might be, for example, the letter S. So this little dog might be little e, little b, little e, little e, little b, little b, little s, little s. So the little e means that it's not making the Phe melanin, and the little b means it's not making the U melanin, and then little s means it's not making the product of the S gene, which I actually don't know what it is. All right, so suppose you want to know more, and so how did I write these slides? I like this website. It's a coat color calculator, and I download it onto my cell phone, and I can, they have all the genes there. They have remember I showed you A, B, C, D, E, all those genes. They have all the genes in there, so you can toggle them for which allele Ma gives, and then you go to the next screen, you toggle it for which allele Pa gives, and then the next screen gives you what the offspring is likely to be. So that one is kind of cute. All right, any questions before we move on?
What is the website like link?
So I usually just Google stuff. See this is the company, Le Dog Co, LEDOGCO, Le Dog Co, and then coat color calculator. Got it? Yeah. All right. So I just sorry I didn't put the link in. All right, so I have my next story. So my next story is the story of Dr. Mary Dawson and the Alpha Wolf, and it comes to you from 1974 when I was a young teenager. So Dr. Mary Dawson is a scientist that I work with very briefly. I was 14 years old, and I loved science. Back in 1974, there weren't that many women scientists, and there weren't that many girls going into science. So I was very lucky, and my parents found me some mentors to help me out. And Dr. Mary Dawson was a cool vertebrate paleontologist. So she studied fossils that had backbones. Her area of interest was the Arctic region. She studied things like mice and other rodents, and she also studied rabbits and hares, Arctic hares. And this is her on her one of her Arctic expeditions. And she was also a curator at the museum where I lived. So I went to the museum, and she handed me a box of just looked like sand and rocks. And there were little fossils in there. And she said, go wild, find the fossils. And I was supposed to find little tiny, tiny jaws of tiny, tiny mice that had lived long, long ago. So and at lunchtime, she told stories that spooked me, and I want to share one of these with you. So she was an Arctic researcher, and she would plan an expedition. She would get people together, she would get supplies together, and the airplane would drop them off, and the area was covered with snow. So she had to pack everything. Nobody was going to rescue her. You had to be very well planned. And sometimes it was very snowy and cold. And as summer got hotter and hotter, the snow melted, and it was easier to find the fossils. So here they are looking for fossils, and they are shaking out snow and sand to try to find the little jawbones of what they were looking for. Interestingly, there were also Arctic wolves present, and sometimes the wolves would watch them. it was almost like the humans were entertainment for the wolves, and they would see these white wolves like watching them. So one day, Dr. Mary was out. She just had one other person with her, a man, and she was breaking the trail for them it was still snow. They both had backpacks on. And so she was leading the other scientist with their backpacks on. They had their lunches in there. They were going to a new site. They were moving from their tents to their site. And Jim behind her said, "Mary, there are some wolves following us." And she said, "I know." So they kept walking, and the wolves were walking along, an entire pack of wolves. So I don't know. I don't recall how many, 7, 9. I don't know how many are in a pack. So they were in a line, and their leader was breaking snow for them. And then they came up alongside Mary and Jim. And Jim said, "Mary, we left our guns back in our tent, and we don't have anything to scare them off with." And she said, "I know, Jim." And she just kept walking, and the wolves kept following them. And she said, it was just like there was a bunch of dogs with them, because they were like trotting along next to the humans. And but then the line of wolves came closer and closer to Mary and Jim. And Jim said, "Mary, the wolves are getting closer. What do you think we should do?" She said, "Let's drop our packs. Maybe they want our peanut butter and jelly sandwiches." So they dropped their packs, and they kept walking. The dogs went up and sniffed the packs, and then the lead alpha wolf came running up to them. And so Mary didn't know what to do. The wolf ran up to her. He ran in front of her, turned around, jumped at her face, and his nose went right up to hers, like a dog kisses you. It went up to her, and the feet didn't actually touch her. It twisted in the air and went back to the ground. And then it trotted off at right angles to Mary and Jim and went off. It was the alpha of the wolf pack greeting the alpha of the human pack. So that's my wolf story. And there she is, Dr. Mary Dawson, when she was much older, a cool Arctic paleontologist and museum curator. Any questions or comments about Dr. Mary? All right, so I want to talk a little bit about saving the wolves of Isle Royal, which is a national park in the state of Michigan. And you can see that the wolves here have fur that's called agouti, and their agouti fur has the 2 pigments we talked about before. They have the B gene and the E gene. The E gene is giving the black color to the overall fur of the wolf. And then the pheomelanin, the E gene, is giving 2 patches of, in this case, it's sort of an orange. So then each hair on a wolf is both black and red. And so that it's called an agouti pattern. So our species here is the gray wolf, and it has the genus name Canis and the species name Lupus. The gray wolf gave rise to dogs twice, and dogs were domesticated from wolves, according to what we know about genetics, a couple of times, about 10,000 years ago. So we think just like the wolves were associating with Dr. Mary and Dr. Jim and kind of hanging out and watching them, we think that twice humans managed to encourage dogs to work with them, maybe hunt with them. And so there's an Eastern dog progenitor and a Western dog progenitor. Then humans started breeding dogs for various traits in order to help humans hunt or to herd their sheep, for example. And that led to changes in the genes. And dogs became a new species, and the genus name is still Canis, but the species name is Familiaris, because we're familiar with them or used to them. All right, so in the chat, do you want to tell me which is your favorite park? Do you have a state park or a national park that's your favorite? You can write that in the chat. I love the national parks, and I love the state parks that are often right next door. Okay, Isle Royal or Grand Canyon. All right. Isle Royal and Arcadia. Yosemite. Oh yeah, these are all great parks. I have pictured here Isle Royal, and my family and I went recently for the first time. So I want to tell you a little bit about the wolves of Isle Royal. And so Isle Royal, if you don't already know it, is located up here in Lake Superior. It is north of the Upper Peninsula. And so here it is. It's a 45-mile island, and you either have to take a boat or you have to take an airplane in order to get there. Wolves crossed over the ice to Isle Royal in 1949 or 1950. And so here's one of the wolves of Isle Royal. The food chain over in Isle Royal consists of many, many pounds of vegetation. And eating that vegetation includes one mammal of interest here, the moose. So we have that pictured here. So there are many fewer moose pounds than there are pounds of vegetation. And then at the top, we have the wolves who eat the moose, and there are even fewer pounds of wolf than there are pounds of moose on the island. And so this is called a food chain with a lot of biomass at the plant level, less biomass at the primary consumer level, the moose level, and then the least biomass at the consumer level, the wolf level. Here's the food web that they tell you about when you go to Isle Royal National Park. So here are all the primary producers or photosynthetic plants. And then here are the primary consumers that eat the plants. And at the top are the secondary consumers. And so the wolf integrated itself into the food web. So a food web is a bunch of food chains all put together. And the food chain that I showed you is right about here. Wolves control the moose population. And if the moose population are not controlled, then the moose eat up all the vegetation. And shown here is the number of wolves and on the left, and the number of moose on the right, and the year along the bottom. And what we see is a corresponding rise and fall of the species. So let's take it from left to right here. In the left and the dark blue, we have the moose population rising because they're reproducing and there's lots of good vegetation. The wolves that eat the moose population then starts to increase because they're eating the increasing number of moose. Their population increases and they end up eating the moose, so the moose population decreases. And this pattern of the moose going up first, and then the wolf population going up second, and then the moose population falling, and falling of the wolf population repeats. So the populations of them go up and down, not exactly in sync, but one is delayed compared to the other. Now, climate change has changed the breeding patterns. Initially, the wolves were getting additional wolf genes because occasionally a wolf could cross on ice. But climate change made warmer winters, breeding with the mainland wolves stopped, and then there was inbreeding between the wolves on the island, and the wolves became more and more related. So they were breeding with closer and closer relatives. Like first they were breeding between fifth cousins, and then fourth cousins, and then third cousins, and then second cousins. And this inbreeding led to defects in the wolf offspring, and that included crooked spines, extra vertebrae, and ribs. And so they weren't as healthy as they had been. By 2017, they were down to a breeding pair. It was actually a father and a daughter. They were highly related, and they couldn't have puppies. So the population of wolves was going to go to zero. I'm showing you a zero with my hand here. And so this picture is showing a pack of wolves. I guess they're all wolves. Yeah, a pack of wolves running in the snow, taken probably from a helicopter. So the solution was to move or translocate 19 gray wolves in order to increase the population and the diversity. So trapping wolves can be tricky because they can be pretty smart, and they wanted to get a genetic diversity. So this is a conservation effort. So they trapped them from around Michigan, from Canada, Minnesota. They wanted to get gray wolf populations over a large geographic area because most wolves breed with, mate with whoever's nearby. So they picked a wide area to get the wolves from. So they anesthetized the wolves after trapping them. They checked them for their teeth. Here's a wolf coming out of the cage. So it must be terribly scary to be dangling from the bottom of a helicopter. Then the cage lands, then the helicopter lands. Then they open the cage, they let the wolf out, and the wolf moves away from the helicopter. And the question is, how successful is this effort? And it's working. And here we have healthy wolf puppies being born. So that's pretty cool. Are there any questions or comments about wolves? Arctic wolves, gray wolves, there are a large number of different kinds of wolves. We've just looked at 2. Okay. Does anyone know the Game of Thrones direwolf ghost? Familiar with that one? All right, I put that picture in because I figured some of you might have seen the movie or read the books. And now I'd like to show, oh, sorry, go on, Max.
Just on Royal, wasn't there also like the moose population got like there were a lot of moose because of the black and wolves and
Exactly. So
trampling stuff.
the moose were eating all the vegetation. So
Yeah.
it really changes the way the island functions when you have overabundance of the primary consumers. Yeah,
Okay.
yeah, you got it. So you can do this lab along at home. You can't do it right now, but I'm going to drop the link in the chat of how to do it with the list of supplies and how to do it. And I'm going to show you a video of me doing it at home during the pandemic to show my students in college how to do this simple experiment. So it's an extraction of DNA from strawberries, and the key ingredients are you have water, you have strawberries, you have salt, and you have rubbing alcohol, which is a poison. So you have to do it, you have to get your parents to get you the supplies and give you the permission. And it's recommended to protect your eyes, wear glasses or sunglasses and keep the rubbing alcohol away from children and pets, including your dog. This is the supply list, and I wasn't sure if anyone wanted to take a photograph. I'm going to drop it in the chat. All right, everybody's good. Okay, and so let me check the video, and I think I may have to stop sharing and select the video. Okay. Share screen with the video. All right, tell me, raise your hand or something if you cannot hear the sound. It should work. In order to do this experiment, I have a list of supplies for you below, and you can follow along with me. You can turn your video on and off as we go. It's going to be pretty fast though. The first thing you're going to need are your 3 strawberries, and you're going to need to hull them, which means you need to pull off or cut off the green part here. I've actually hulled mine using a knife. I find that easier. So you're going to have 3 of those. You're going to put them into a resealable plastic bag, and this bag, you're going to burp most of the air out of it and seal it up. It is helpful also if you already have your rubbing alcohol in the freezer. So mine is frosty cold, and I've already made my extraction liquid in order to get started. So in an old olive jar, I have placed a half teaspoon of salt, a 1/3 cup of water, and 1 tablespoon of liquid dishwashing soap, you know, the kind for hand washing dishes. All right, let's get started. You put those 3 strawberries in a bag and you mush them for 2 minutes. You can turn off your video. when you come back, your extract from strawberries should look approximately like this. It's pretty messy. Now we're going to actually get the DNA out of the cells. So we've broken up the strawberries, but a lot of the cells are still intact. The cells are surrounded by lipid membranes, as is the nucleus. So we have to get through 2 sets of membranes to get the DNA out. We're going to put in 3 tablespoons of our extraction liquid. Again, that's half teaspoon of salt, a 1/3 cup of water, and a tablespoon of dishwashing liquid. We're going to put 3 tablespoons in. And at this point, I have an apron in my lap in case this gets messy. So there's 1, 2, 3. And now in real time, I'm going to mush this. Whoops, I'm going to push some of the air out. There we go. And mush this for 1 minute. Oh, your kids will like to do this. All right, we're breaking open the cells. We are making what's called a lysate. When we break open cells, we are lysing them, L-Y-S-E. This is a Greek word. And at this point, the DNA is starting to flow into our solution. We have salt in the solution, and the DNA is binding to the salt, and this is going to help us precipitate it out at the appropriate moment. All right, so new use for dish soap here. I have that about a minute has passed. I have placed a filtering solution. I've got another spice jar. In it, I've placed a funnel, and I have cheesecloth in there, or you can fold up a coffee filter and open up just 1/4 of that. I'm going to use scissors or a knife to snip my bag, and I'm going to pour out the liquid, and hopefully not too much of the still solid strawberries. And so I'm going to squeeze this through. And what we're doing is we're pulling out all the large cellular matter, strawberry bits, seeds, and cell matter. And we're basically straining it through the cheesecloth. Right, and the instructions are to, I don't know, fill up this jar maybe a 1/4 full. We're getting pretty close. All right, I would say that's close to a 1/4. Next, I'm going to take my icy cold rubbing alcohol, and it asks for 1/2 cup, but we may not need all of that. I'm going to layer in on the top. I'm going to layer in 1 inch of rubbing alcohol. In order to do this, I'm going to tilt both of my jars and gently pour it in. Let's see if I can see this. I got, all right, I have about 1 centimeter. Maybe I'll put more in. All right, it is pouring in my lap. And now I'm taking my stir stick. Oh, I can see my DNA. I'm going to take my stir stick and dip it down and pull the DNA up and try to precipitate it in the alcohol. There it goes. Trying to wrap it on my stir stick. Oh, I'm getting it. It's just very large amounts of DNA. Very cool. It is very sticky. And I'm trying not to mix my 2 layers. I have tried to wrap it around my stick. It's a little hard to see, but my stick is getting nice and thick. Now, if you're very good at wrapping this, I'm used to precipitating it out using a centrifuge in the lab. So I don't know if you can see, I'm starting to get lumps of the DNA on here. Wow, just so much DNA. And we would precipitate this out in the lab. When people do DNA extracts for any purpose, for paternity tests or for DNA sampling to determine if the correct fish have been caught on board a vessel, all that kind of DNA testing, forensics and otherwise. This is the type of test that is used. We often do it in very small test tubes and then again spin the DNA out. I hope you've enjoyed our DNA extraction lab. See you next time. All right. So does that look like a technique that you would be able to do at home and complete? Get yourself some rubbing alcohol. All right. And I will just go back to the PowerPoint. I don't think I have anything left, but this is the DNA. So I had my husband take a picture of me with it. So the DNA, there's a drop of water, but honestly, it looks like snot. It's kind of clear and white, and it's very exciting. You can get really big wads of this DNA. So that is a pretty cool thing that can be done. I'm hoping that you will stay curious about your world, and thank you so much for attending. I'd love to take any questions. Oh, thank you. Thank you, Vicki. Do you folks have any questions? Did you want to see my stuff? I already showed you my stuffed animals. So this is the dog, the stuffed animal dog from my kids' room. My daughter just moved out last week. And then I got these wolf puppy dog stuffed animals from the Detroit Zoo. The Detroit Zoo has a mating pair. They have a male, a black male, and they have a white female. It's not actually white, a gray female, a black male and a white gray female. And then the zoo down in Toledo also has one mating pair. And it's pretty exciting. At Washtenaw Community College, our logo involves we're called the Wolf Pack. So our spirit animal that gives us spirit at basketball games and stuff is a wolf. So we're the Wolf Pack, and so we're very fond of wolves.
Well, thank you, Lawson and Max and Vicki for attending today, and thank you, Emily, for taking time on a Saturday to share some interesting information about the genetics of wolves and dogs. That was really interesting. And I love the stories that you include.
Thank you. Thank you so much. I forgot. I just put the instructions. Oh, thank you so much. I put the instructions here in the chat. So if you know how to use Zoom and how to download from Zoom, let's see, what do you do?
Emily.
Oh, I have to send it. There we go.
All right. Emily, Lawson mentioned too, Winter Dance by Gary Paulson. Have you heard of that book?
I haven't. Do you want to tell us about that?
I have not actually read it. Max has though. I've never read it, but I just know it's about the Iditarod and how he had he raced it, and I don't think he actually ever won, but
he did it twice.
Yeah.
He's just talking about like the dogs, and is it like he doesn't control the dogs control. He's just like a guy on a sled, and the dogs know where to go.
Yeah, that's the sense I was getting. My husband said, well, how did they find their way through the storm? Did the person driving the sled, did the musher have a compass? And I said, no, the reports are the dog is going home to its kennel, and it just goes straight there. It can smell things. It can hear cracking ice. It can smell and hear and sense which way to go. And they just went absolutely arrow straight back to their kennel.
Wow. Wow.
Yeah.
Very, very, very interesting, but it makes me feel cold thinking about it.
I agree with you.
Yeah, he said that like dogs operate best at like -20 because he lived in Minnesota. I think he still does.
Oh,
Yeah, it makes sense.
I think these sled dogs had adaptations. They have a double coat. I'm not sure what a double coat is. I'm assuming a coat and maybe a really thick undercoat. So they have some extra fancy way to survive in the cold that like our regular dogs couldn't survive in that
I
cold.
have 2 bunnies and they also have it. It's like an undercoat that's like kind of goose down. It's just really soft and it's meant to hold heat in, and then the overcoat is like meant to shed water and not get them dirty. So it's kind of like 2 coats.
Wow.
Yeah.
Wow. And then a lot of animals too have, you know, adaptations in their feet to keep their extremities warm, and yeah, it's pretty amazing. We don't have those adaptations as humans. We just have to put on all of our things and build shelters and
I think I also heard for the Iditarod, you travel with a large number of dogs, and sometimes the dogs are on your sled, and then you change them out and reharness them.
much
Yeah,
like Canadian geese, right? Where they switch positions and
a little bit, yeah. But that might have been like more modern because like the sled where you was talking about like the photos in the book, like the sled wasn't really that big because they had checkpoints and you'd ship ahead like your gear, so you wouldn't carry everything the whole way.
Mhm.
interesting. Well, everyone, enjoy your Saturday, stay warm, and I will put this recording up on our website too, our Super Steam website. And Max and Lawson, I hear that you're coming to the dinosaur, the Dinosaur Super Saturday in February with David Wooten. Is that
Yeah.
right? Yep,
Mhm.
yep. So we'll look forward to seeing you then. And thank you again, Emily.
You're so welcome. Have a good Saturday, everyone.
Bye.
Thank you.
Bye. Thank you.
You're welcome. Bye-bye, everyone.